Today, we live in a world where the tiniest invisible fragments of plastic are not just polluting our oceans, but infiltrating the very building blocks of life, our genes, our brains, and our futures. Today, I'll take you on a journey through this invisible threat, the charged nanoplastics reshaping the story of neurodevelopmental disorders. I am Minji Kim from Asia Pacific International School, and this is a story we cannot afford to ignore. First, I'll be explaining the correlation between nanoplastics and neurodevelopmental health. So, what are nanoplastics exactly? Nanoplastics can be defined as synthetic polymer particles that are even smaller than microplastics. Their minuscule size allows them to maintain a bigger surface area. The increasing surface area to volume ratio formulates an electric charge as UV radiation breaks the structure of the plastic, ultimately allowing them to acquire a surface charge. This allows different environmental toxins such as lead or bisphenol A to attach themselves onto the nanoplastics, and these harmful toxins may pose severe risks to both human health and the environment. Nanoplastics are already known to impact and cause different neurological disorders such as Alzheimer's and ADHD. Going deeper into neurological disorders, I wanted to investigate whether nanoplastics could also potentially cause negative impacts on neurodevelopmental disorders as well. As of recently, gene biomarkers that are representative of different developmental disorders have been identified. To be specific, gene mutations in ZFX have been expressed in ADHD and autism spectrum disorder, or also known as ASD. Mutations in KDM6B were also recorded in patients with ASD, while most patients with Down syndrome show an overexpression in the gene DSCR1. As developmental disabilities have already indicated patterns of gene biomarkers, the possibility that exposure to nanoplastics could impact the level of gene expression can now be measured through their impact on such biomarkers, which could mean the discovery of the potential for nanoplastics to exacerbate these developmental disorders. Ultimately, the purpose of the experiment was to measure whether nanoplastics can penetrate the human cell, investigate the harm nanoplastics can bring depending on their positive or negative surface charge, and then observe whether exposure to nanoplastics could indirectly impact different developmental disabilities. From now on, I'll be explaining my materials and methods. First, bits of plastic were put into a flask with DW and shaken on a rocker for a month. The plastic and DW mixture was then extracted and observed under the inverted microscope to see whether nanoplastic was actually present in the mixture. After checking its presence, the nanoplastic mixtures went through electrolysis to split between positive and negative surface charge. Afterwards, the split nanoplastic mixture went through electrophoresis to check the presence of the different positive and negative surface charge. Second, human neuroblastoma cells were cultured with DMEM medium as seen in Figure 3. In Figure 4, cultured cells were treated with fluorescent nanoplastics for 1 hour, 24 hours, and 48 hours to check penetration. Fluorescent nanoplastics were used to observe penetration as general nanoplastic. Being transparent wasn't able to be depicted in the cell. However, cells were then treated with the positively and negatively charged nanoplastics to use for future gene analysis. The RNA of cells exposed to nanoplastics were extracted, and cDNA was synthesized with the gene primers related to the developmental disorders. After, gel electrophoresis was conducted to compare the gene expressions. 4 different biomarker genes were used, and the following primer sequences can be shown in Table 1. From now on, I'll be explaining my results. Figure 6a represents the observation of general nanoplastics dyed using Nile red under the inverted microscope. Figure 6b represents how both green and orange fluorescent nanoplastics showed a strong attraction to the negative pole, meaning the majority of fluorescent nanoplastics had a positive charge. In Figure 7, you can see that the nanoplastics successfully penetrated into the cytoplasm of the human neuroblastoma after 24 hours. The nanoplastic particles inside the cell's cytoplasm were observed under the fluorescent microscope even after washing 3 times. Because although nanoplastics were proven through my experiment to penetrate the human cell after 24 hours, the results would be futile if they were unable to pass the blood-brain barrier. Nanoplastics must be extremely small in order to enter the blood-brain barrier, and although my specific mechanics of how it enters the brain are not yet determined, my experiment was supported by further recent cases last year by Yuhan Ma that evidenced that nanoplastics indeed are able to pass the BBB. Both DNA and RNA are negatively charged, so when charged nanoplastic, especially those of positive surface charge, enters the nucleus, it may prevent the combination of RNA polymerase and DNA, or the combination of RNA and ribosomes, which could potentially lead to abnormal RNA transcription. To see whether the phenomenon could actually occur, I observed the RNA expression of cells already exposed to nanoplastics. Figure 8a shows the intensity bands of both RNA and PCR product of GAPDH. In total RNA, while the control band is observed to have two bands, the band exposed to nanoplastics with a negative surface charge has a single yet thicker band. Meanwhile, the band exposed to nanoplastics with a positive charge has two bands and is slightly thicker than the control band. In 8b, the band intensities were measured as numbers. In contrast to 8a, the control bands that were not exposed to nanoplastics and bands that were exposed to positive surface charged nanoplastics represented similar values, while the band exposed to nanoplastics of negative surface charge was represented as a bigger value. Specifically, the value of the negative band was increased approximately 1.3 times the control value. In GAPDH PCR product, the negative nanoplastics and positive nanoplastic bands decreased in comparison to the control band. In Figure 8c, it shows that while there is a difference in negative nanoplastics and positive nanoplastics with intensity of 0.60 and 0.81 respectively, both intensities were smaller than the control variable of 1. Both decreased approximately 20% in respect to the control. The results differed from the results of the RNA because the same amount of RNA was used. When dividing the intensity of GAPDH by the intensity of total RNA, the intensity ratios of negative and positive nanoplastics were calculated to be 0.6 and 0.8 respectively. The smaller ratios of negative and positive bands indicate abnormal decreased transcription. Therefore, it suggests that the nanoplastics have also affected biomarkers associated with different intellectual disabilities, implying that nanoplastic interference alters the proportion of RNA while the proportion of protein remains unchanged. I postulated that there might be two potential mechanisms behind the phenomenon of abnormal RNA transcription. First, a direct impact on RNA, which holds a negative charge, with a nanoplastic's positive charge, or second, disturbing a pathway from the cytoplasm to cause an indirect problem in the RNA. The fact that exposure to nanoplastics causes abnormal RNA transcription indicates that this may also indirectly impact the genes of developmental disorders. In this experiment, 4 different types of gene biomarkers were used to see whether they were impacted by nanoplastics. As seen in Figure 9, CFX, a biomarker representative of general neurodevelopmental disorders, showed an increase in expression after exposure to positively charged nanoplastics. KDM6B, a biomarker representative of ADHD and autism spectrum disorder, and DATA1, a biomarker representative of ADHD only, both showed an increase in expression after exposure to all charged nanoplastics. However, DSCR1, a gene biomarker representative of Down syndrome, showed a decrease in expression after exposure to both positively and negatively charged nanoplastics. I posited the reason for this outlier result to be because Down syndrome is in fact a genetic disorder caused by an abnormality in the 21st chromosome and is not a developmental disorder. Last but not least, the total impact of nanoplastics on the expression of developmental disorder biomarker genes was evaluated by multiplying the intensity of biomarker expression by the RNA transcription impact. As shown in Figure 10, in all groups except for DSCR1, representative biomarker of Down syndrome, positively charged nanoplastics increased the intensity of abnormal gene expression compared to the negatively charged nanoplastics. However, the lower expression intensity observed in the negatively charged nanoplastic exposure group compared to the control group also appears to be an abnormal result. In conclusion, the harmful interference of nanoplastics penetrating cells within only 24 hours suggests the potential to impact the expression of genes associated with various developmental disorders, including but not limited to autism, ADHD, and Down syndrome, therefore potentially promoting the onset of these disorders and exacerbating their symptoms. These microscopic invaders may be silent, but their impact on our future could be deafening. Thank you.